Okay, let's talk fundamentals. Where does everything alive actually get its energy? To move, to grow, just to be alive?
That's the big question, isn't it?
It really is. And you, our listener, sent over some fantastic material that digs right into this. Really fascinating stuff.
Yeah, we got this PDF, Cellular Respiration by Mr. Chi, and it's great diagrams, experiments laid out. It really focuses on how organisms unlock the energy they need.
It's such a core process.
Absolutely fundamental.
And that process is cellular respiration. That's our focus for this deep dive. It's like this tiny, powerful engine inside every single cell, turning fuel into usable power.
Exactly. A microscopic power plant.
So our mission today is to get a real handle on it. Using the insights from the material you shared, we'll look at the main types, how they work, and where we see them in action.
Sounds good. Ready to jump in?
Let's do it. So maybe the simplest starting point. What exactly is cellular respiration in basic terms?
Okay. At its core, it's a chemical process. It happens in living things, and it's all about breaking down complex molecules, think sugars like glucose, to release the energy stored inside them.
Breaking things down to get energy out.
Precisely. And the material you sent introduces some key ideas around metabolism here. Metabolism is kind of the umbrella term for all the chemical reactions keeping an organism going. Within that, you've got catabolic reactions. Those are the breakdown ones, like respiration. And then anabolic reactions, which build things up.
Got it. Catabolic is breaking down, like cats knocking things over. Maybe not. Anabolic is building up. Respiration is definitely catabolic.
Right. It's taking that fuel molecule apart.
And why is that released energy so critical? I mean, it seems obvious, but the material spells it out.
It does. This energy powers pretty much everything a cell needs to do. Cell growth, making new cells through division, any kind of movement.
Like muscle contractions.
Exactly. Or even moving substances across membranes inside the cell, especially active transport, which needs energy to pump things against their natural flow.
So no energy release, no life, basically.
Fundamentally, yes. None of those core processes could happen.
Okay. Now, the material makes it clear this isn't just one single reaction. It talks about aerobic respiration first. That sounds like the main one.
It often is, especially for larger organisms like us. Aerobic respiration is the kind that absolutely has to have oxygen present.
Needs oxygen.
And it's super efficient at getting a lot of energy out of, say, a glucose molecule. The diagrams in your resource show where this happens.
Yeah, I saw those. Looks like it starts in one place and moves somewhere else.
That's right. The first step, glycolysis, happens out in the main body of the cell, the cytoplasm.
Okay, the jelly stuff.
But the next, much bigger energy-releasing stages, the Krebs cycle and oxidative phosphorylation, those happen inside the mitochondria.
Ah, the famous powerhouses of the cell.
Exactly. That name really fits, especially for these later stages. Their internal structure is actually really well suited for maximizing this energy production.
Makes sense. So let's break down the stages. Glycolysis first, out in the cytoplasm. What's happening there?
So you start with glucose, a 6-carbon sugar. Glycolysis basically splits that one molecule into two smaller 3-carbon molecules.
Cutting it in half, essentially.
Sort of, yeah. And this initial split releases a little bit of energy directly. But maybe more importantly for the next steps, it releases these things called high-energy hydrogen ions.
High-energy hydrogen.
Yeah, think of them as carrying energy-packed electrons that used to be in the glucose bonds. They're like little batteries, ready to be used later.
Okay, little energy carriers. And the material mentions this step doesn't need oxygen?
Correct. Glycolysis itself is anaerobic. It can happen whether oxygen is around or not. That's a key point.
Interesting. So glucose gets split, we get a tiny bit of energy and these energy carriers. Then things move into the mitochondria for the Krebs cycle. But this needs oxygen, right?
Yes, the Krebs cycle won't run without oxygen being available somewhere down the line. It happens inside the mitochondria, in the matrix, the sort of inner fluid, and it's this series of reactions, like a cycle, that takes those smaller molecules from glycolysis and breaks them down even further.
Getting more energy out.
Well, it releases more of those high-energy hydrogen ions, definitely. Yeah. And it also produces carbon dioxide. That's a waste product here. Plus, it regenerates molecules needed to keep the cycle going.
Like a little chemical roundabout, churning out CO2 in these energy carriers?
You can think of it that way, yeah. And that leads us to the big finale. Oxidative phosphorylation. Quite a mouthful.
It is. What's the payoff here? This is where the main energy comes from.
This is the big one, yes. It also happens inside the mitochondria, but more on the inner membranes. And this is where oxygen directly comes into play. It's where almost all the ATP is made.
ATP. That's the actual energy currency the cell uses.
Exactly. Adenosine triphosphate. Think of ADP as a partly charged battery and pi as a loose phosphate group. Oxidative phosphorylation uses the energy from those hydrogen carriers to stick the pi onto ADP, fully charging it to become ATP.
How does that work with the hydrogens?
Those high-energy hydrogens, or really their electrons, are passed along a chain of proteins in the membrane, like a tiny bucket brigade.
Okay.
As the electrons move, they release energy. And that energy is used to pump protons, hydrogen ions, across the membrane, building up a gradient, like water behind a dam.
Right, creating potential energy.
Exactly. And then that gradient is used to power an enzyme, ATP synthase, which actually makes the ATP. It lets the protons flow back across the membrane and harnesses that flow to make ATP from ADP and pi.
Wow, that's intricate.
It really is. And then finally, those electrons and protons, having done their job, need somewhere to go. That's where oxygen steps in. It accepts them and combines with them to form water.
Ah, so oxygen cleans up at the end.
It's the final electron acceptor. Without it, the whole chain backs up, and ATP production via this route stops.
Which explains why oxygen is so vital for this whole aerobic process, and the material boils it all down to an equation.
Yep, a neat summary. Glucose plus oxygen gives you carbon dioxide plus water plus a whole lot of ATP.
Yeah.
Show the inputs and outputs clearly.
Glucose plus O2, CO2 plus H2O plus ATP. Got it. That's aerobic respiration. High yield needs oxygen. But what if there's no oxygen? That's anaerobic respiration, right?
Correct. Anaerobic means without oxygen. And as the material highlights, you get way less energy out of glucose this way because you miss out on those big ATP-producing steps in the mitochondria.
So less bang for your buck, energy-wise.
Definitely. The text mentions two main kinds you'll encounter: alcoholic fermentation and lactic acid fermentation.
Alcoholic fermentation. Sounds familiar. Like in brewing.
Exactly. This happens in yeast and also some plants and other microorganisms. Glucose gets broken down, but instead of going through the Krebs cycle and everything, it ends up as ethanol. That's the alcohol and carbon dioxide.
And just a little ATP.
Just a little from the initial glycolysis step mainly. But the byproducts are hugely important, as your resource points out. The CO2 makes bread rise.
Ah, the bubbles in dough.
Yep. And the ethanol is, well, the point of making beer and wine. It's amazing how we've harnessed this microbial process.
It really is. Bread and beer from the same basic pathway. Wild. Okay, what about the other one, lactic acid fermentation?
That's the one that happens mostly in animal muscles, especially during really intense exercise.
Like sprinting.
Yeah, exactly. When you're going all out, your muscles might be using energy faster than your bloodstream can deliver oxygen for the full aerobic process.
So they switch tracks?
They rely more heavily on glycolysis. And then to keep glycolysis going, they convert the pyruvate, that molecule, from splitting glucose into lactic acid. Again, it produces a bit of ATP, but not much.
And lactic acid. That's the stuff that makes your muscles burn.
That's the theory, yeah. The buildup is associated with muscle fatigue and that burning sensation. Your material also notes that some bacteria do this too, and that's vital for making yogurt and cheese. Gives them that tangy taste.
Huh. So muscle burn and tangy yogurt, same basic chemistry.
Different context, same pathway. Lactic acid fermentation.
Okay. So the material includes a table comparing aerobic and anaerobic side by side. That sounds useful.
Yeah. Table 1. It's a good summary. Key differences. Oxygen needed for aerobic, not anaerobic, products. CO2 and water for aerobic, ethanol and CO2, or lactic acid for anaerobic. Location. Mitochondria, heavily involved in aerobic, mainly cytoplasm for anaerobic. And the big one, energy yield much higher for aerobic.
Really lays it out clearly. Okay, now seeing this in action helps. The resources describe some investigations, like experiments. Let's look at the first one, proving oxygen is used.
Right. Investigation 1. They suggest using germinating beans. Seeds that are starting to grow are really active, respiring a lot.
Makes sense.
So you put these germinating beans in a sealed test tube with some lime water. Lime water turns cloudy or milky if carbon dioxide is present.
Okay.
You also set up a control tube, maybe with beans that have been boiled so they're dead and not respiring.
Got it. Compare living and non-living.
Exactly. The idea is, in the tube with the live beans, they'll use up the oxygen inside the sealed tube for aerobic respiration, and the CO2 they produce should turn the lime water milky. The control tube shouldn't show those changes.
So it shows oxygen consumption and CO2 production linked to the living beans.
Well, this one's primarily aimed at showing oxygen is used, maybe by measuring the oxygen levels directly or inferring it. But yeah, the CO2 is often observed too.
Okay. And the second investigation focuses squarely on the carbon dioxide.
Investigation 2, yeah. Similar setup, germinating beans, lime water. The main goal here is just to show that the lime water does turn milky, proving that CO2 is definitely produced during aerobic respiration by the beans.
Simple visual proof. Okay, what about proving CO2 comes from anaerobic respiration? There's one with yeast, right?
Yes, investigation 3 looks at yeast doing alcoholic fermentation. You mix yeast with sugar solution in a test tube, no air, ideally, or very limited.
The sugar is a fuel.
Right. Then you connect this tube with a delivery tube leading into another test tube with clear lime water. Maybe keep it warm to help the yeast work faster.
Okay.
And you'd have a controlled, maybe just sugar solution, no yeast. In the tube with yeast, because they're fermenting the sugar anaerobically, they should produce carbon dioxide gas.
Which bubbles through the tube.
Into the lime water, turning it milky. The control tube stays clear, shows the CO2 came from the yeast's anaerobic activity.
Nice. Making the invisible gas visible. And the last one involves a snail and pondweed. Seems a bit more complex.
Investigation 4, yeah. Uses snails in water with bromothymol blue, that's another indicator, like lime water, but it turns yellow when CO2 levels increase.
Okay, bromothymol blue goes yellow with CO2.
Right, so you have snails in this solution. They respire aerobically, using oxygen, releasing CO2, but then you add variables. Some tubes are kept in the dark, some in the light with pondweed added.
Why the pondweed and light dark?
Because pondweed photosynthesizes in light. Photosynthesis uses carbon dioxide and releases oxygen, the opposite of respiration.
Okay, so it complicates things.
Or rather, it illustrates the interplay. In the dark, the snail respires, releases CO2, indicator turns yellow. The pondweed also respires in the dark, adding more CO2.
Right, no photosynthesis to counter it.
But in the light, the snail still respires, producing CO2. But the pondweed is photosynthesizing, using up CO2. So the indicator might not turn yellow or might turn less yellow, depending on the balance between respiration and photosynthesis rates.
Wow, so it shows the snail's respiration, but also how another process, photosynthesis, affects the gas balance in that little ecosystem.
Exactly. It demonstrates respiration in the snail, but also the interaction between organisms and processes affecting CO2 levels.
That's a really cool demonstration. Okay, so we've seen the processes, seen how to demonstrate them. Let's loop back quickly to the practical uses. We mentioned bread and booze from alcoholic fermentation. Any others highlighted?
Well, the material definitely re-emphasizes those, maybe mentioning traditional brewing methods too. But it also strongly points towards lactic acid fermentation's role in the dairy industry.
Yogurt and cheese again.
Absolutely. Specific bacteria fermenting the milk sugars, producing lactic acid, that's what causes the milk proteins to change texture and gives those products their characteristic sharp flavors. It's a huge industrial application of anaerobic respiration.
It's really everywhere when you start looking, from exercise fatigue to food production.
It underpins so much. These basic energy pathways are just fundamental to life and human civilization in many ways.
So wrapping things up, cellular respiration, it's how life gets its power, comes in two main flavors. Aerobic, needing oxygen, super efficient, makes CO2 and water.
And lots of ATP. Happens largely in the mitochondria.
Right. And anaerobic, no oxygen needed, less energy payoff, makes ethanol and CO2 or lactic acid.
Used by microbes for fermentation and by our muscles in a pinch.
And we saw how experiments can demonstrate these gas exchanges and how fermentation is key for making things like bread, alcohol, yogurt, cheese.
It connects the microscopic chemistry to everyday life.
Definitely. So, reflecting on all this, we see organisms using these different strategies. Some need oxygen constantly, others thrive without it. It makes you wonder, doesn't it? Well, thinking about an organism's main energy strategy, aerobic versus anaerobic, what could that suggest about, the kind of place it originally evolved in or the environment it lives in now, its whole history?
That's a great question. The environment definitely shapes which pathways are favored or even possible.
And thinking about those industrial uses, bread, beer, yogurt, are there other ways we might be using or could potentially use these fundamental biological engines, maybe in areas we haven't even thought of yet?
It's certainly possible. Harnessing biological processes is a huge area of innovation. Definitely something to chew on, the hidden influence of cellular respiration all around us.